Now that we've discussed the retina and how processing occurs there, we can discuss the movement of the information from the retina back to the brain. The axons from the ganglion cells we discussed are leaving the eye through the optic disc. And axons going from one location to another can be referred to as projections. And when we say there are 3 retinofugal projections, we mean there are 3 projections away from the retina. Namely, a little less than 10% of the axons are going to a brain region called the pretectum in the midbrain. This is important for pupillary responses and not particularly interesting, so not one that we'll really discuss. The second one is the tectum, part of which is the superior colliculus, and around 10% of the projections from the retina go to the tectum. And this region is important for saccadic eye movements, it's more sensitive to peripheral stimulation, and is related to the movement of attention. And then the bulk of the retinofugal projections from the retina go to the cortex, and this includes higher order visual processing as well as conscious vision. So let's talk about the second one, the tectum, and in particular the superior colliculus. As we said, this region is important for saccadic eye movements. Saccadic eye movements are just basically the typical eye movement. If you move your eyes from left to right or up or down, you're using saccadic eye movements. And the superior colliculus has neurons that are particularly important for moving the eyes to specific locations. And the receptive fields are somatotopically organized. That is, where in the visual field the superior colliculus neurons respond is linked to where that location is relative to the body. So soma here being body, and topically being topology, so somatotopically organized. Now, there are 7 layers to the superior colliculus, and the super layers, or superficial layers, receive direct input from the retina and also receive indirect input from visual cortex. These neurons in the superior colliculus are particularly interested in the periphery of the visual field rather than the center. So that foveal processing we talked about, that's mostly going to visual cortex. The superior colliculus is not so interested in the fovea, it's more concerned with the periphery. Now, why would that be? Well, let's take note of the way this structure looks. The superior colliculus is down here in the midbrain. Midbrain is evolutionarily older than the cortex that surrounds it. So this brain region presumably developed earlier in evolution and is related to more primitive visual functioning. More primitive visual functioning would be centered on identifying threats in the environment. And usually threats are not coming head on toward you, they're coming from the periphery, from your sides. If somebody's trying to sneak up on you, they're not going to walk straight toward you, they're going to try and sneak up from the side. And so your superior colliculus is mostly about detecting what's going on off to the side. Okay. And this is very good for survival, and makes sense that this is an evolutionarily older brain region. So those superficial layers, as we said, receive direct input from the retina. The deeper layers actually receive some direct input from auditory cortex as well as somatosensory cortex. So not only is the superior colliculus receiving information about the visual field, it's receiving information about the body and about hearing. And this makes sense because if, again, to take the example, someone's sneaking up on you, if they're sneaking up beside you, maybe they're making some noise, and that will help you localize, that'll help you move your eyes right to where that person is. Okay, so you want auditory information to inform the saccadic eye movements that the superior colliculus is making, and maybe that person is close enough to be touching you, they're touching you, say, on your shoulder, then you want to be able to use that body sensation also to help guide your saccadic eye movements to that location so you can identify whether this is a threat as quickly as possible. If a little less than 10% of the retinofugal projections go to the pretectum, about 10% go to the tectum, then that leaves a little over 80% of the retinofugal projections to head toward visual cortex. But they don't go straight to visual cortex, they're going to make a stop first. So let's talk about these retinofugal projections to the visual cortex. As we said, these retinofugal projections are bundles of ganglion cell axons that are exiting the eye through the blind spot, which is that optic disc. And those ganglion cells bundle together and form what we call the optic nerve. Next is going to be the optic chiasm. These optic nerves, these ganglion cell axons, some are going to cross over at the optic chiasm, whereas some are going to go straight back. So chiasm means X, or crossing, and there's a crossing here in the visual system. Right here is the optic chiasm. Let's look at a picture of it. If we look here, we're looking at the bottom of the brain. We've got the frontal lobes up here. We've got the temporal lobes here, the brainstem and the spinal cord, cerebellum. Well, right underneath the frontal lobes is the optic chiasm. Now, in this particular image, the optic nerves have been severed, but if they hadn't been severed, the eyeballs would be connected to them and would be right about here. So as we said, some of the information crosses over the optic chiasm, but some of it goes straight back. To understand how this works, we want to first note that it's not that the left eye goes to the right hemisphere. Students often get mistaken into thinking that. It's not that the left eye goes to the right hemisphere, nor is it that the right eye goes to the left hemisphere. It's that the left visual field goes to the right hemisphere, and the right visual field goes to the left hemisphere. Okay, so it's a very important distinction. Why? Because each eye sees both the left and the right visual field. So that means if we consider the left eye, it is seeing the left visual field, and that information coming into the left eye from the left visual field needs to cross over because it needs to make it to the right visual cortex. However, some of the information coming into the left eye is coming from the right visual field. And the right visual field has to end up in the left visual cortex. So information from the right visual field entering the left eye needs to not cross over. It needs to go straight back. Okay? So some of the information coming into the left eye is going to the right hemisphere. That's left eye information that's coming from the left visual field. But some of the information entering the left eye is coming from the right visual field and therefore is not crossing over at the optic chiasm, but rather is going straight back. So again, it's not that the left eye goes to the right hemisphere, it's that the left visual field goes to the right hemisphere. So there are not axon terminals in the optic chiasm. There are just the axons passing through. Where do those axons? They terminate in the lateral geniculate nucleus. The lateral geniculate nucleus. This is a region of the thalamus. The thalamus is a structure near the center of the brain that has about 9 different nuclei, and one of those nuclei is the lateral geniculate nucleus. So the optic nerve axons are terminating in this lateral geniculate nucleus, and they're terminating in 6 different layers of the lateral geniculate nucleus, labeled 1 through 6. Now, 3 of those layers, layers 1, 4, and 6, are receiving contralateral input. So if it's the left lateral geniculate nucleus we're talking about, then layers 1, 4, and 6 are receiving input from the right eye, whereas layers 2, 3, and 4 are receiving ipsilateral input, or same side input. So again, if it's a left lateral geniculate nucleus, then layers 2, 3, and 5 are receiving input from the left eye. So ipsilateral versus contralateral. But there also is another distinction that you can see based on the darkness of these layers. Layers 1 and 2 are stained darker than layers 3, 4, 5, and 6. And this is because layers 1 and 2 are receiving magnocellular input. Magno means magnanimous or magnificent, means large. So these are larger cells with larger axons that are coming into the magnocellular input. The way this image is created with this dye in it is the dye is injected into the eye, and the animal shown as stimulus, and then the dye makes its way back to the lateral geniculate nucleus. Since magnocellular cells are larger, they're going to transport more dye. Therefore, we see them as darker spots in layers 1 and 2. The lighter spots in layers 2, 3, 4, 3, 4, 5, and 6 are lighter because they're parvocellular, smaller cell size, and therefore they do not transmit as much of the dye. Okay, continuing with the lateral geniculate nucleus. One thing to note is this is the first place in the ascending visual pathway that is sensitive to feelings and arousal. One interesting thing about the visual system, and this is probably true, honestly, of the other sensory systems as well, but its processing is modulated by how you feel. So the way you see the world around you is related to the way you feel about the world around you. Therefore, it's no surprise that early in the visual processing stream at the lateral geniculate nucleus, we're already seeing the visual processing happening in the lateral geniculate nucleus being influenced by your feelings. There are intermediate and deep layers that contain somatotopic, tonotopic, and retinotopic maps. So we talked about the superior colliculus having somatotopic and auditory or tonotopic input, and then retinotopy is important there as well, meaning that the way the cells are arranged in lateral geniculate nucleus is determined by the way the cells are arranged in the retina. The topographic organization of the retina is preserved in the lateral geniculate nucleus. Now let's talk about the lateral geniculate nucleus sending its axons to primary visual cortex, or V1. Specifically, if we look over here at the lateral geniculate nucleus, we can see that almost all of the input from the lateral geniculate nucleus is going to layer 4 in primary visual cortex. In particular, layer 4 has several sublayers to it, and the sublayers that are coming to the lateral geniculate nucleus are in the 4C alpha and the 4C beta layers. For our purposes, it's just good enough to know that LGN axons are going to layer 4. As we mentioned, the topographic organization of the retina is preserved in the input of the lateral geniculate nucleus. Well, this is true in V1 as well. Retinotopy is preserved. So we've gone from with the blind spot out the optic nerve to the optic chiasm, and talked about some of the axons crossing over, some of the axons going straight back. These are going to go to the lateral geniculate nucleus where the axons will terminate and synapse with dendrites in the lateral geniculate nucleus. Those dendrites will receive excitatory postsynaptic potentials, depolarizing the axons of those lateral geniculate nucleus neurons to excitation threshold. And where are those neurons going? They're going to V1, or primary visual cortex. So let's talk a bit about how primary visual cortex neurons were first studied. They were first studied by a couple of researchers named with the last names of Hubel and Wiesel, who won the Nobel Prize for their research. As is common in science, many of the most interesting findings happened somewhat by accident. Hubel and Wiesel knew that there should be visual neurons in the occipital lobe, but they wanted to be the first ones to go and record from these neurons to confirm this hypothesis. So they put their electrodes in individual neurons in the primary visual cortex. That we can see, by the way, back here in the posterior part of the brain. Most of the primary visual cortex is in the medial portion of the occipital lobe along this calcarine fissure. Calcarine fissure, an important term here. And we see that on the fissure is just a larger sulcus. Okay, and so we can see this sulcus or this fissure running along the occipital lobe in the medial portion, and there's an upper bank of that sulcus and the lower bank of that sulcus. That's where primary visual cortex lies. So Hubel and Wiesel had individual electrodes implanted into individual neurons in the primary visual cortex, and then they used an ophthalmoscope to shine light on the retina of the monkeys that they were studying. And what they expected is that when they shined this little dot of light onto the retina of the monkey, they expected to see the electrodes activate because the neurons in primary visual cortex would activate. But this didn't happen. They'd shine a light in one location, nothing happened. They shine it in another location, nothing happened. They shine it at every point on the retina, and nothing was happening in this primary visual cortex. So they decided, well, let's change the size of the dot that we're shining on the retina. So if you recall your physician using an ophthalmoscope, the ophthalmoscope has a dial on it that can make either a small circle of light or a large circle or anywhere in between. So they started adjusting the size of that spot of light, and that wasn't helping the neuron fire at all. Except on one trial, the ophthalmoscope jammed. It was halfway in between two different sizes of spots of light. And instead of creating a circle of light, that created a semicircle with a straight edge on one side of the circle. And all of a sudden, the neuron started firing vigorously. And what Hubel and Wiesel discovered, because this ophthalmoscope jammed, they discovered that primary visual cortex is interested in not just light, but in borders of light or edges. Remember that we talked about ganglion cell doing edge detection. Edges are very important to the visual system, and we see even back in primary visual cortex that these neurons are primarily interested in edges. So let's talk about the receptive fields of these primary visual cortex neurons, these V1 cells. There are three kinds: simple cells, complex, and hypercomplex. Simple cells care about the orientation of the line in their receptive field. Complex cells care both about orientation and direction of movement. Hypercomplex cells care about orientation, direction, and the length of the lines. Although current research suggests that hypercomplex cells might not be their own class of neurons, but rather it might be that some simple cells and some complex cells are sensitive to length. These are called end-stopped cells, and simple end-stopped cells and complex end-stopped cells. But you need not worry about this last point. For our purposes, we'll just consider there to be three different types of V1 cells: simple, complex, and hypercomplex. Now let's just talk about the first two, simple and complex. This image over on the right is trying to illustrate the properties of simple cells. If we take the blue rectangle here as the receptive field of a neuron we're recording from, we can look at these vertical lines and consider those action potentials in that primary visual cortex neuron. So, for the particular neuron we're recording here, if we shine a light of this orientation in the receptive field, the neuron fires vigorously. As we move the light, the edge of light or the bar of light into a different orientation, the neuron responds less and less again until when the bar of light is oriented in the opposite direction, this particular V1 neuron will not fire at all. This simple cell simply cares about the orientation of the bar of light. Now, while this neuron prefers orientation like this and does not respond at all to an orientation like this, you could find a different V1 neuron that would respond maximally to this orientation and not at all to the opposite one. And you can find neurons, if you keep looking around, you'll find neurons that respond to each of the 365 degrees of orientation. So simple cells care simply about the orientation of the line. This image on the left is trying to describe what happens in complex cells. They also care about the orientation of the line, and in this example, we're going to just assume we've got a neuron that likes a 90-degree vertical orientation. That line, it's not enough for that line, though, to be of perpendicular orientation. It needs to be moving in a direction from left to right. So what this image is indicating is that when this bar is set in motion, when it moves into the receptive field, the neuron fires vigorously. And once the bar has exited the receptive field, the neuron goes back to baseline levels of firing. Take the same bar of that same orientation, but move it from right to left, and the neuron barely fires at all. So complex neurons in the primary visual cortex care not only about the orientation of the line, but also its direction of movement. So we have simple, complex, and hypercomplex neurons in primary visual cortex, and some of these neurons are grouped into blobs, as they're called, cytochrome oxidase blobs. And they're so named because they absorb cytochrome oxidase stains. And they're called blobs because, as we can see over here to the left, they've got a kind of amorphous shape. They're not nice structured columns or modules. They're blobs. These cytochrome oxidase blobs receive input from parvo and coniocellular layers of the lateral geniculate nucleus. We didn't mention coniocellular neurons when we discussed the lateral geniculate nucleus, but these are the layers that are in between the parvo and the magnocellular layers. Cytochrome oxidase blobs process information from color-sensitive ganglion cells. They are very important for color processing, and they're found primarily in layers 2 and 3. We can see them illustrated here, layers 2 and 3, and more faintly, not quite as robust of blobs, are found in layers 5 and 6 as well. So these blobs are connected from V1 to V2. We can see here that the cytochrome oxidase blobs are inputting to a layer of V2 referred to as thin stripes, whereas the regions, interblob regions, the regions outside of the blobs, have neurons that are projecting axons to V2 pale stripes and thick stripes. So we can see over here, blobs project to thin stripes. Here's the thin stripe. Therefore, this is information about color. Interblobs project to thick and pale stripes. So we've got thick stripes here, pale stripes here. And this is projecting information about orientation, spatial frequency, movement, and retinal disparity. Now, an important feature of visual processing concerns spatial frequency. Hubel and Wiesel showed that primary visual cortex neurons were sensitive to lines. Later researchers found that V1 actually responds best to sine wave gratings, which is what we see over here. A sine wave is, of course, just a smooth oscillating line, and we can envision a sine wave as alternating black and white bars. And if the alternation, if the borders between the black and white bars are smooth, then these are considered sine wave gratings. And V1 cortex seems to really respond strongly to these sine wave gratings. And it's the orientation of the lines in those sine wave gratings that simple and complex cells, as well as hypercomplex cells, are sensitive to. To understand sine wave information, we can look at these two images on the right. The two pictures contain the same amount of low frequency information, but extraneous high frequency information has been filtered from the picture on the right. So the picture on the left has a lot of high spatial frequency information. High spatial frequency information is indicated by sharp edges. So these pixels have sharp edges to them. And it's quite interesting, if you can walk back from your computer screen far enough, then this image on the left starts to look the same as on the image on the right, because from a further distance, this high spatial frequency is not resolvable by your eyes. Therefore, the high frequency information goes away when you look at it from a distance, and all you see is the same thing that you see here on the right side of this image.